I want to welcome everybody to tonight's Honor Symposium lecture. I'm Jane Karras, president of the college, and thank you all for all of your continued support for the college, because without this great community, we wouldn't have this wonderful college to provide education and learning opportunities for people here in Northwest Montana. This year's Honor Symposium topic is technology and humankind, and we feature a variety of expert voices that will discuss the benefits and dangers of technology and offer a balanced view about its impact as we inevitably interface with technology in most aspects of our daily lives. We will examine technology's implications on nature, our environment, the self, the family, and society. We will explore if technology helps or hurts humanity. We will investigate some of the promises and perils that humanity faces now that the technology genie is out of the bottle and here to stay. In just a minute, I'm going to introduce our second speaker of this thought-provoking series. Please enjoy tonight, and please join us as we welcome a great lineup of speakers in the following weeks. You all should have one of these programs that will give you the list of the next 4 speakers, and hopefully you will be able to join us on those dates. I want to thank Gerda Reeb for organizing this wonderful series of lectures, and then recognize our generous Honor Symposium sponsors, the Theodore Chase Endowment Fund, the American Association of University Women Kalispell Chapter, a grant from Humanities Montana, and the FVCC Foundation Alumni and Friends. So thank you all for making this Honor Symposium possible. Thank you. It is my pleasure tonight to introduce Michael Goguen. Michael devotes his time and energy to making positive impacts on the lives of others. He is a philanthropist, a venture capitalist, and a first responder with Two Bear Air, the search and rescue operation that he founded and funds at zero cost to taxpayers and rescue victims. Following a 30-year career as a leading technologist and venture capitalist in Silicon Valley, Michael now resides full-time in Montana. His focus is on finding solutions to local and statewide challenges to improve the lives of Montana residents, and on helping entrepreneurs start and build new and innovative companies that could have positive effects on everyone. During his 20-year career at Sequoia Capital, Michael appeared frequently on the Forbes Midas list of the world's top venture capitalists and led approximately 50 early and growth stage technology investments, resulting in a combined market value of nearly $50 billion. Prior to Sequoia, Michael spent 10 years in various engineering research and product management roles at DEC, Synoptics, and Centillion. Michael was also a technical chairman of the ATM Forum. He has a bachelor's degree in electrical engineering from Cornell University and a master's of science degree in electrical engineering from Stanford University. Before we begin, I would just ask you, at the end of the talk, we will have time for questions, so please no interruptions during Michael's talk. At the end, we will have 2 mics to hand around. I would ask you to keep your questions brief and to the point in order to allow everyone to have a chance to ask a question that would like to do so, and to please keep your questions tonight focused on tonight's topic. So please help me welcome tonight's presenter, Michael Goguen.
Well, thank you. You know, who in the audience has had to deal with an incurable illness, either that you faced yourself or you've gotten a diagnosis that's pretty bad, and doctors tell you they'll give you what they can, but you know yourself that there's really no cure, or even worse, a child, or if you are young, a parent or a brother or a sister. Since humankind began, we've been frustrated with not having the answer we want when we hear that kind of news or face that kind of situation. It affects every single one of us. And for me, in the latter part of my Silicon Valley career, the most rewarding part of what I ever did was to get involved in precision medicine cancer companies because they seemed to have some kind of progress. They were making progress. And I was very honored to be part of a few that were very, very successful for what they were able to do. But it was also one of the most frustrating experiences of my life because when you get deep into the science, and we'll get a few inches deep tonight, you realize that something like cancer, which affects a large percentage of the people in this room, I think one way or the other, and many people around the world, at its core is such a deep right at the core code of life kind of disease. You know, our DNA, the programming of every single cell in our body going haywire, and our cells doing something they're not supposed to do. That goes right to the core of it. And the frustration that all companies had, and I was feeling too when I was involved in these companies, is we just don't have the ability to do anything about what we can see. We can start to see what's going on, but we have no ability to do anything about it. So when gene editing first started becoming a topic, and there was a buzz about some new revolutionary gene editing technology that was coming about, all of us were pretty hopeful. And boy, we just didn't know what we were in for, because the gene editing technology that's happening around the globe at a just a viral pace has implications way beyond the positive implications that myself and thousands of others were hoping for disease treatment, which it certainly does. It is incredibly powerful. But it goes beyond that, and I'll talk about that tonight. We as humankind, having been on this planet, having been on this planet for billions of years, but humans only for 60 million or so, have the very first time we have the ability to change the code of our life, of evolution. We have to change the code of any living thing. We can make species extinct. We can change the properties of any species on planet Earth very simply and for a few $100s, as I'll explain. And that's both thrilling and frightening. I'm going to talk about both of those, but in terms of having an impact on the rest of your lives, I think you'll see it. You might not hear the techie terms, but you'll see the ramifications of it, whether you care about human health, curing disease, the environment, agriculture, anything else that's going to have a direct impact on this valley, and probably already is in little ways that you don't quite realize yet. My wife used to say that this reminds her of the story of Prometheus, the Greek god who handed humankind fire, and Zeus was really mad because he said, humankind isn't ready for this. Well, I don't know if we're ready for this or not, but it's here, so we better be. When I talk about the source code of life, we talk about being able to edit the source code of life. Most of you, especially if you're not a biologist, don't have much frame of reference. You might not even be a technologist. So the closest you come is the device that seems to rule your life these days, the iPhone. And you have this vague understanding, perhaps, that deep inside there's a bunch of ones and zeros, and that's the source code. And that is true. About 70 years ago, humans discovered the transistor, or invented the transistor, made this little 1 and a 0 with electricity. And 70 years later, we have this magical device that obsesses us all that can do some pretty cool things. And if you look at it, that seems pretty thrilling, that you can look at those little set of 1 and 0s, because that's all that computer starts with. There's no written sentences in there when you turn on your iPhone. It looks at those little 1s and 0s and somehow does all these cool things and gets you obsessed with social network and everything else. Seems pretty impressive, right? But nature has us beat by a long shot. So nature's had about 3.5 billion years to work on what they did with their little code, what nature did with its little code. And as it turns out, in the late 1800s, it was discovered that every single form of life on planet Earth, be it the smallest little bacteria to human beings, to whales, to fish, to jellyfish, when you look inside their cells, they all seem to have a common molecule, and it was this very strange molecule. It's kind of the coolest molecule on Earth, called DNA. Long, stringy-looking thing, looked like a ladder, kind of twisted. You've all seen the pictures probably, not really understood what it meant. But it's just this long, funny-looking molecule. But it's in everything, the same molecule. And boy, it seemed to be important because, for example, with human beings, when we start as a single cell, it's about the only molecule in the cell. A couple other chemicals in there, but that's it. So talk about something pretty cool. This molecule must somehow be able to not only do all the cool functionality that we do as a grown-up human, as in an iPhone can do when it reads its code, but it creates us. It self-assembles us somehow from that code. So there really must be something going on there. So we knew DNA was important since the 1800s. It was in the 50s that scientists discovered that it actually had a code. Now, this won't be on the test. You don't have to memorize this. But the point of it is just to understand that there really is a code. Now, it's not ones and zeros. It happens to be 4 letters. And these represent different chemicals that make up the rungs of the ladder. And it has this nice, simple little set of rules as to which letters can go with which. Turns out to be really important when cells replicate and DNA wants to replicate itself. You only need half the ladder, because if you know the rule, every letter you see, you know what the pairing is, so you can make another half. Again, not on the test. But the point is this. That's a code. That really is a code. And it's the same code for every single living thing on Earth, every blade of grass, every human being, every whale in the ocean. Now, it's packaged differently, so not to be confused, when many of you hear about genetics and genomics and everything, you think about those funny X's, the chromosomes, right? Here's put it in reference, then we won't talk about chromosomes the rest of the night. But human beings happen to have their DNA super tightly packed in these funky looking things called chromosomes, which then get even more tightly packed in the nucleus of every cell in our body. Okay, that's these X shape. We have 23 of them. So our DNA is actually cut into segments and squished into these things. Something like a bacteria just has it as one big ball of spaghetti. It's actually looped, but its DNA looks the exact same as ours, just packaged a little bit differently. Now, DNA is different lengths for different creatures. We actually have a little over 3 billion rungs on our ladder. So in other words, we kind of have 3 billion instructions in our little instruction set that make us human. Bacteria has a little less. A couple of little factoids to tell your friends about DNA, by the way, at cocktail parties. So think about a human being having 10 trillion cells in your body, roughly. And every cell is way tinier than the head of a pin, right? And the pointy end. But in that cell is DNA. And there's enough DNA in that cell that if you strung it out, it'd be about 6 feet long. So if you actually took all the DNA in your body, those 10 trillion cells, you could go around the sun and back 100 times. So anyway, it's kind of very efficiently encoded, if you will. Now here's the part, it might look a little technical, but this is really critical to understand. I'll try to keep it real layman's terms. It isn't just the code that's common between everything in life. It's actually the way the code is interpreted. And what we discovered not too many years ago was that, hey, first it was just a bunch of letters. We named these letters and said, great, we have this code we can't understand. Then we started to notice some patterns. And one pattern was there are these certain symbols, sequence of letters that seem to define these little beginning of a sentence, end of a sentence. And the stuff in between we named genes because that set of codes seem to do the really important work in DNA. Now the workhorses in a cell or in life itself actually are proteins, and it's not what most of us think of. Proteins as the shake you take to get all buffed up after you work out. Proteins actually are one of the most complicated molecules on Earth. There's tens and hundreds of millions of different varieties of proteins, and proteins have these really funny properties when they're made. If you mix certain ones together, they'll make a muscle fiber, and mix another couple together, and they'll make eye color. They'll do all kinds of different things. So the really big discovery was in the beginning of genetics was, hey, there are these codes, and they do really important things. And you've all heard, for example, you have the gene for brown eyes, or you have the gene for blue eyes. So some genes actually encode directly a function, brown hair, blonde hair, and so on. Others are a little more subtle. They'll encode some really important protein that you or I wouldn't know the name of, but it turns out if your cell is producing that protein in the right amount, and maybe another gene on your DNA in that same cell is producing the other protein at the right amount, the cell's going to be healthy and do what it's supposed to do. If they're not, if one of them's not producing it, the way they mix causes something really bad to happen. So every single one of these proteins is super, super important. Humans have about 25,000 genes on those 3.5 billion instructions, which actually, when you do the math, says that's only about 10% of those instructions. And for years, genomists, leading scientists called the rest of it junk DNA. They said, oh, it's left over from evolution. We don't really know what that is. They're starting to think differently. And part of the reason is we've discovered the other important thing. We have a gene, and this is all relevant to what we can edit and what we'll talk about. And then we have these factors. I call them the volume control. So that's not the technical term. That's the way I think about it, and it's helpful for you to understand it. The technical terms you'll hear are gene expression, gene regulation, epigenomics. We won't talk about that much. We'll stick with my layman's terms, volume control. Because here's what happens. It turns out the genes are important, but genes can be on or off. In fact, they can be really active, like cranking out a lot of that thing they're producing, or they can be not very active at all. And it turns out, as a critical component in human health, it's gene expression, sometimes more than what the instruction says, what the actual DNA says, that we've now found out is just as important, if not more, and also related to a huge variety of diseases. So the way this kind of works, and here, by the way, is the solution to the is it nature or is it nurture problem. This puts it all together, by the way, for all of you folks who've been wondering, well, what the heck did I read 10 years ago about broccoli being good for me and not good for me? How does that cause cancer? I thought it's all my genes. Here's the answer. So your genes are what they are, and you might have ones that are in code for something that Maybe we'll do something bad or good, that's fine. But the way the volume gets turned on and off, there might be part of the volume control in the cell, but a lot of it comes from outside. So here's where your environment comes in. You have an unhealthy diet, unhealthy exercise, and so on. Those molecules that are coming from the outside of the cell might not be so good. And that's what causes genes to get turned on in funny ways or turned off. So for example, let's take something very specific. You had one of those 23andMe tests and it says, oh my gosh, you have a gene that says you're high risk for breast cancer or some kind of cancer. Well, it doesn't mean you'll get cancer. Whether you will or won't depends on whether that gene gets turned on or off, or stays off. Hopefully it stays off. So that's where your environment comes in. Okay, so anyway, this kind of puts it all together. Now we have the volume control, and you can see the volume control kind of sticks to the DNA. Literally, the molecules do. Sometime really sticky, and sometime transient. So in other words, some genes are turned on off all the time. Others get turned on or off, and they stick that way. And that causes, for example, some fairly permanent conditions when we get into the medical stuff that one wishes we could correct, right? So once scientists figured this out, that we have genes, they can code for different things, and they can turn on and off, we started to realize that not only a lot, but I would claim nearly every human ailment that causes death, permanent disability, ruins your life, all relates to what's going on your little code, your DNA in your cells, in terms of what the genes are doing or not doing. Because what we've now found is, first of all, there's the obvious ones. Over 16,000 inherited genetic diseases. That means you had an error in the gene. Some kind of mutation happened, luck of the draw, or your child, unfortunately, was born with the letters the wrong way, right? So instead of making the protein that was supposed to do something wonderful for their body, it does something terrible. In fact, tragic, oftentimes. So there's some horrible, horrible diseases that are mistakes in the code. And boy, we wish we could correct that, right? Didn't have the ability. 300,000 viruses. I was astounded by that. There are 300,000 different viruses that affect mammals, right? And as you'll see in a super cute pictorial I have, what viruses do. Viruses' whole job is to screw up your DNA. Viruses literally get into a cell, and what they do, how they cause problems, is they kind of blow up your own DNA, and I'll show you how. So that's sort of a genetic related issue, right? Here's some of the shocking ones that are very, very recent. Psychiatric illnesses, maybe even addiction. I'm actually certain addiction is. They're related to genes that get stuck on or off in the wrong positions. Boy, don't we wish we could do something about that, right? Heart disease, and then finally the big one, cancer. Cancer, by definition, is a disease of the genes being mutated, screwed up, or stuck on or stuck off in the wrong way. So scientists were desperate to do something about it. Now we'll get into the good, cute pictorials that I took a long time, and apologize if the animations are pretty crude, but it was my first time. I will tell you one thing. So that's supposed to be a virus, right? And here's the part that creeped me out a little bit. There's a real virus that looks like that. Looks like a little spaceship on an electron microscope. So if you want to know what a virus looks like, that's kind of what at least one of them looks like. So what do viruses do? Viruses grab onto a cell, they get in your bloodstream, right? Viruses are these weird creatures. We've never been able to tell whether they're really alive or dead because they don't actually have cells. They're these weird little pieces of genetic material, seem to have the mind of their own. They carry some genetic material, but this is what a virus does. A virus injects its own DNA and just jams it into your DNA, right? You know, it's really interesting. Remember that 3.5 billion instructions we have? Scientists now believe that several hundred million of those instructions are remnants of prehistoric viruses that have jammed its own DNA into our DNA. So each of us are carrying around that in each of our cells. But here's what the problem is. This is the purpose of them jamming DNA. They don't do it just to be troublemakers. That DNA starts producing like crazy more little copies of the viruses, and you can guess the next step, the cell blows up. So that's when we get sick. That's the flu that just went around, the flu virus. That's every virus. That's what's happening in our cells, okay? Now, scientists desperate to find a way, how can we start manipulating this code of life, you know, focusing, of course, on humans and diseases. They looked at this and said, boy, that's kind of clever. You know, viruses, I wonder if there's a way to hijack a virus, and maybe there's a good gene we want to stick in this sick patient who doesn't have the right gene. Maybe if we can just jam a good one in there, something good will happen. So I'll just remove that. They started using viruses as delivery mechanisms. In fact, when you first heard of gene therapy, That's one of the main mechanisms they were using. There were a few others. But that was it. They were literally taking viruses, they're making them less dangerous, they were kind of twiddling them around a little bit, sticking the healthy gene, and then injecting you with that virus. And here's the obvious problem based on how you saw the mechanism work. Kind of sometimes it worked, and that gene they were sticking in there went in the right place, good things happened. Sometimes it didn't work so well, right? In fact, there were really devastating clinical trials on a fairly high-profile drug years back that was supposed to do something really wonderful. And in the trials, it gave some people leukemia, a deadly form of leukemia that they didn't have before. And that was because it's this brute force method, that material, those instructions were getting jammed in there, causing all kinds of damage. So this wasn't the perfect mechanism. So we were desperate for a real solution. The breakthrough came from who I believe will become one of the most famous scientists of all time in humankind, Dr. Jennifer Doudna, 2012 at University of Berkeley, California. She was a molecular biologist, wonderfully brilliant woman in all kinds of the right areas, and she was studying these kinds of mechanisms. And here's the breakthrough that led to this gene editing that we'll talk about the implications of. It's called CRISPR-Cas, technical name. You don't have to really understand why. I'm not going to go describing the techie terms behind it. I will show you how it was discovered because it's pretty darn fascinating. So it turns out bacteria are infected by viruses too. Those infect the little spaceship virus. It comes from one that happens to blow up bacteria all the time, which you think might be a good thing, but there's a heck of a lot of good bacteria that we don't want blown up. So viruses still to me equal bad. Anyway, so the viruses would be attacking this bacteria, but here's what the scientists started noticing. In a lot of species of bacteria, they seem to be kind of immune to these viruses. Somehow the viruses, they could see it attacking the cell, nothing was happening. Then they started looking at the bacteria's DNA, and they noticed something weird. First of all, there were these repeating patterns. That's where CRISPR comes in. It's this long word that means something about repeating. But see, the gray areas are supposed to represent this same letter code. It would repeat for literally thousands, if not hundreds of thousands of sequences. So that's odd. Here was the real observation. All of a sudden, some scientists realized, hey, wait a minute. The DNA that this virus is trying to inject seems to match exactly, see the purple DNA between the gray? And that bacteria seems to be resistant to it. So every time there was a cell, bacteria that was resistant to a virus, it always seemed to be the case that somehow in its own DNA, it had encoded a little fingerprint of the viruses that it seemed to want to, that it could somehow protect against. And they said, that's odd and that's fascinating. Let's figure out how this works. So this is how it works. So, I told you molecular reactions are always kind of reading DNA all the time. Same way in every species, as I told you about, reading the proteins, start, stop, all that stuff. So even in bacteria, there's little chemical reactions that are always reading the DNA and producing things, right? Well, as it turns out, what gets produced from this little structure here is there's this little thing called a Cas up there. That produces one molecule, pretty complicated molecule. It seems to fuse with the little pattern match molecule. Then, it kind of floats around in the cell. Then the virus goes to inject its DNA. This thing seems to snatch onto it like a magnet and blow it up. And we were fascinated, right? So they dug in further and they found out that the sort of orangey part acted literally like genetic scissors, seemed to snip it precisely. So the little lock-on pattern locked right onto the pattern, and the scissors snipped it precisely right there. They said, wow, but here's the brilliant breakthrough. All of a sudden, Professor Doudna and her team said, wait a minute, have we just discovered the answer to what we've been looking for? What if we turn this around? What if we engineer that same little molecular package, a little pattern match, a little scissors? They were able to do this in the lab. We're able to do this now, recreate things synthetically. And then what if, let's say that was a human cell. What if that was a sick gene? What if that was a gene that caused sickle cell anemia? What if that was a gene that caused cancer? What if that was a terrible gene that caused a horrible thing? What they engineered and have proven successfully and now is literally blown the world's mind, and I can tell you about all the implications of what's happened since, this one paper that proved they could do this, is that they could get this into a cell. What would happen is the target would lock on. So now we're talking about not the way the viruses did it. We wouldn't just jam something in there. We would lock on precisely to what we were after, right? the molecular scissors would cut the DNA, and DNA just happened to have this sort of natural property, we already knew this, that it wants to fix itself. You know, if the strands break, it's looking for fixing itself, and if you just so happen to have the right pattern where the ends match, nicely, shazam, you precisely fix that. Yeah, sorry, let me just fix the little animation. You've now fixed the gene. And we went, wow. So since that first innovation, there have been hundreds of tweaks to this system, and it can do an enormous number of things that I can tell you about. But now we have the ability, humankind has the ability to manipulate, edit to a precision degree, just about everything to do with those little codes and the volume controls. That was a new innovation. They modified the scissors part so it wouldn't actually cut, but it would still lock on and it would glom onto the volume control. So now, wait a minute, we can regulate, we can fix genes, we can add in new genes, and we can fix whether they're being expressed or not. The power of that is what the rest of the talk will be about, and what has the world really both in awe and a little bit terrified. One of the reasons that it's both so exciting and terrifying is the other breakthrough in all this is CRISPR. That technology is incredibly simple, incredibly cheap, incredibly easy to use. In fact, this picture is literally of a do-it-yourself kit that's out there that has sold thousands and tens of thousands of people. Now we're using this around the globe, where you can edit genes yourself for a few $100s. You can edit your own genes. In fact, there's an article about whether the FDA should regulate against modifying your own genetics, because there's some people crazy enough on stage or injecting themselves. I think the founder of this company is a fairly eccentric character. I think he did that, and so on. But you can imagine this is causing both the progress to be faster than really any innovation in the history of science, but also the worries. So let's talk about one of the first incredibly important applications that really looks promising. If you think about mosquitoes, you're looking at one of the deadliest creatures on Earth, by the way. A lot of you realize that, some of you don't. The pesky little mosquito that causes you to have welts causes some of the most deaths of any creature on planet Earth based on the diseases they carry, right? So several million deaths a year. I was reading statistics, several 100 million severe illnesses per year. Malaria, dengue fever, yellow fever, and so on. That's all this perfect. And doesn't this remind you of the virus picture a little bit? It's kind of what a mosquito does. If it lands on your body, not your cells, and it squirts in its stuff, and in fact, oftentimes a virus payload. So what scientists started doing is looking at the DNA of mosquitoes and saying, gee, I wonder if I can tweak the gene that lets the virus come in. They're getting down in the technical details, but could there be things they could change in the code so that mosquito just couldn't carry malaria anymore, or dengue fever, and so on? And the answer was yes. So they've proven they can do that. So then the next question was this. Okay, great. What does that matter? I get a few mosquitoes in my lab and I can tweak them. How the heck does that solve the worldwide mosquito problem, right? Well, it turns out something like mosquitoes breed very, very quickly and die very, very quickly, right? So the population can change very quickly, but there was one more problem to solve, which was if I go with normal inheritance, if I fix the mosquito and it's the red one, and you just set it out into the wild, as you can see, my wonderful fix will die out pretty quickly because it's just math. Half the traits come from each mosquito and so on in the left-hand picture. So that wouldn't work. So an innovation was derived that's both powerful, fascinating, amazing, but also leads to one of the most frightening aspects of gene editing, because now this, once something is out there, it can't be taken back. So they invented something called a gene drive. And what a gene drive allows is you literally, in the red mosquito that you've fixed, you also, in its own genetics, in its DNA, you insert a copy of the little CRISPR machine, so that when the mating happens and the DNA is getting together in the normal pattern of deciding whose genes are going to get what, one of them is a little aggressive and squirts out one of those CRISPR machines, goes over, chops up and edits the other one to make sure that new gene is in there. So 100% of the time, you can get that in there. Now, great, this was done for an absolutely worthy, incredibly wonderful application, but you can probably already start to imagine the potential misuse when I talk about some of the more troubling applications of gene editing, especially when we talk about our own species. So this happened. I'm not sure the status of these particular experiments, but I would predict that within the next few years, you're going to see some dramatic differences in the mosquito-borne illnesses in some of the worst places in the world. Let's talk about agriculture, because that's where there's been a long-ranging debate for years now about modifying food and modifying anything that ends up back in our bodies, right? But we need a little reset here for a second. Human beings have been genetically altering, if you will, the food and animals, domesticated animals, since human beings have been around. Here's why. We didn't know anything about genes and DNA and CRISPR and all these other things. But we did know about selective mating, and what were we doing? We were actually doing unnatural selection. We were selecting for the traits we wanted. Now things started to get a little crazy, at least in the food industry, in the 30s. Do you know in the 30s, we started to understand enough about genetics to know, it's mutations. It's random changes to the DNA, some of which just by luck make a plant better, make that food better, that are really the driver. So food companies, starting in the 30s, started bombarding the DNA of food with X-rays to cause mutations, cause crazy mutations, and then they'd basically see which ones seemed to grow. Hey, that one worked, whatever that did, I'm not sure what it did, but that one seemed to work, let's replant that one, and that became our food stock. Now, you guys have already become such genetic experts that you were causing all kinds of errors too, and who the heck knows what was going on that you wouldn't know about for years by that approach. More aggressively than that was what we really think of as GMOs, which tend to be, frankly, using some of those much more crude gene editing techniques, jamming in, splicing together genes with lots of damage, and who the heck knows where it was. And in fact, as you all know, based on some of the real controversial topics that came up in the area of GMOs, they were using foreign DNA. So, let's see if we take this gene from a jellyfish and splice it together with an apple and see if it makes it, more hardier to sunshine and so on. And maybe it did, but who else, what the heck else did it do? So here's the thing to get your head around, and this is one of the reasons why I'm so glad so many people are here to become educated on this topic that really is going to be in your lives, whether you know it or not, because there will be debates. There will be political debates. There'll be societal debates about this stuff. Is it good or is it bad? You should understand, at least in the area of agriculture and food, CRISPR is not really GMO, because what we're doing so far with CRISPR is we're taking the plant's own DNA. So there's an apple, right? And you're looking at the DNA of that apple, that species of apple. You're not adding genes, you're not doing anything. But let's say there's one that's broken in 90% of your apples, you're tweaking it to the way it should be for that pure line. Or let's say there's a gene expression. Maybe some of the best apples in the batch seem to express that gene, turn on that gene more. Well, now we can adjust the volume on that plant. So the FDA has so far ruled this is not GMO, because their version is, hey, you're actually, you're really looking at what the source code of that species, and you're just tweaking it. You're not adding things, foreign DNA, you're not mishmash jamming things together. So I believe that as well. I'm pretty comfortable with that. There are studies out there, experiments, things happening where they're doing it to livestock as well, making more lean beef, more lean pigs, and so on. There's a gene all of us have, actually, that really regulates muscle growth, and they're basically just amping up that gene. So is that Franken cow or Franken pig? I'm not sure, but that's what's happening. The environmental applications, this should be true and near and dear to many of our hearts out here. So the environmental applications are really fantastic with this technology. But again, it could be misused. But the good applications, the one that seem pretty clean in my mind, ethically and morally, some of the real plagues that affect us all, affects our environment. We all know about the Western beetle plague that's affected vast swaths of forest. Jennifer Doudna herself, actually, I saw a video of her just about a month ago. It was ironic because my wife and I were talking about, gee, I wonder if CRISPR could be used to help solve the beetle crisis. And then there's Jennifer Doudna on a tape talking about what applications are you excited about? And she said, well, I think I'm excited about using CRISPR to solve the Western beetle crisis. And she said, we need to start playing with the modifications of the trees to make them more resistant, because they have DNA too, believe it or not. Or the beetles themselves somehow make it so they can't, inject the thing that's most toxic to trees. But there's also been incredibly promising sort of helping to, quote, save the planet kind of ideas that have popped out with CRISPR and have been proven so far. So take biofuels. I was involved in biofuels when I was a venture capitalist down in Silicon Valley. The problem was wonderful ambitions and so on, but most of you cynics have learned that, from a resource point of view, they kind of maybe hurt more than they help. They take a heck of a lot of resources to produce that fuel that's supposed to be environmental, but you just did more environmental damage in the first place. The efficiencies are really low, costs are high, subsidies, blah, blah, blah. They've already successfully tweaked the genetics of algae, for example, to make it 10 or 100 times more productive in biofuels, or converted some form of wheat to produce another element that's critical to making biofuels way more effectively. They've been tweaking genetics to make things produce less waste. They've been tweaking genetics to make plants consume waste. So what if there are toxins in our environment that we could tweak the genetics of some moss to make it need that and want that and thrive with that? The potential is really, really limitless. So there are also the less comfortable applications that people are talking about. So, we all saw the movie Jurassic Park, most of us did, right? Remember the premise? They had DNA that was caught in the resin of some sap, from prehistoric mosquitoes, giant mosquitoes probably, that used to bite into dinosaurs. Well, a couple things wrong with that premise. DNA is an amazing molecule. It's estimated it can last around 1,000 years, not 65 million. So that's a problem. But there really is a legitimate scientist, in fact, one of the earliest scientists working on gene editing, who's kind of obsessed with this idea. You can debate whether this is a good idea or a bad idea. He's taking the genetics of an elephant and is obsessed with creating a woolly mammoth, bringing back a woolly mammoth. I didn't mention on the prior slide, but it is true that one of the things we can do in the environment and in our ecosystem is manage species a little bit better with gene editing. So for example, if we literally could bring back extinct species, modern day extinct species if we wanted to, if that was a good idea. We could actually genetically engineer some of the invasive species to make them less hardy and so on, to reduce their populations. There's a tremendous amount of tweaking we could do. We have almost infinite control. The question is, how are we going to use it? The medical examples, that was the driver in the first place, right? And that's really where the progress has been astonishing. You really can't keep up if you ever get interested in this stuff and feel like Googling that funny-looking breakfast cereal CRISPR word. You'll see a tremendous amount of progress posted probably from the prior day. It's really happening that fast. And it's really stunning. It goes right to what I claimed earlier. If you really think about what's going on in our molecules, it makes sense that just about every disease ultimately translates to something bad happening in our instructions, or the instructions are getting turned off when they're not supposed to, or funky ones are getting turned on. And that's what CRISPR can modify. Therefore, it shouldn't be shocking that we've reversed mutations in hereditary blindness. We've reversed mutations to permanently cure sickle cell anemia, made living cells impervious to HIV/AIDS, taken out AIDS from a patient that had AIDS. Now, when I say patient, a lot of this is, most of this, 90% of this is laboratory, human cell lines, or maybe mice and so on. There have been some human examples, but obviously we have to go with the regulatory pace. But these things are incredibly promising. In fact, I should have mentioned, in terms of progress on human disease, one of the slightly more Frankensteinian applications, but worthy, is you all know we've used mice for many years and pigs for organ donation. We've used CRISPR now to make their systems even more human. So there are literally cells in mice now, in laboratory mice, that are identical to human cells. And there are organs being grown in pigs that are literally genetically identical to human organs. So even in terms of advancing science, before we get to really stick this stuff in humans or try it, we're able to make tons of progress. Huntington's disease, antibacterial resistant antibiotic resistant bacteria superbugs. We've engineered ways to make them genetically kill themselves after a few iterations. So even edited a gene causing halting and reversing of Alzheimer's. That speaks right to one of my big theses as I've gotten into this over the past few years, is that a heck of a lot of the disease just has to do with what gets turned on and off, because Alzheimer's is not actually a mutation. It's just one of these stuck on or stuck off switches that we're able to play with. Cancer, of course, is the big one. Cancer is, just for those of you who don't know, you've probably figured it out already from my words or known yourself. Cancer is one of those diseases that is 100% related and caused and symptomatic of those two mechanisms I talked about, the volume control and the instructions themselves, right? So you've all heard about mutations in cancer. Well, when you look at a cancer tumor and you do a biopsy of those cells, I mean, it looks like chaos. The cells themselves, actually, there's a lot of mutations, not just one, and the volume controls are all over the place. So there's been a question for a while, which comes first, chicken or egg a little bit? Does somehow the volume control in a weird gene, let's say the one that's supposed to reproduce, tell you how fast to reproduce the cell, does that get stuck on? And then just by statistics, it's reproducing so fast, starts creating a lot of mutations, or is there more going on? Some of the details are still being worked out, but we know it's related to these things. So there's already been tremendous progress. I didn't list them all, it would have been several pages of studies where they've halted cancer of different types, liver cancer, pancreatic cancer, and so on, in lab animals and so on, live creatures, reverse tumors, all using CRISPR-Cas. I think we're around the corner from really having a shot at getting rid of this disease. The human trials are beginning. So let me explain this real quick, because this is what you'll see. And if, unfortunately, any of you have relatives or yourself that face this horrible disease in the near future, relatively near future, this is your first hope. So, getting this little machinery inside some cell that's deep in your body to fight some tumor you have buried in an organ, I think that's possible. We're heading there. But that's just by your own logic, that's a little more complicated, right? Than the way we have been doing it, which seems really powerful. We have an immune system. We're not like the bacteria. We don't have to have it built in our DNA. We have separate cells that run around and fight things, right? T cells is the main one. That's the main cell of your immune system. They're pretty awesome. They recognize just about anything foreign and they kill it. And it's always been a mystery. Why can't they recognize cancer cells? And the answer is it always looks a little bit too much like yourself, right? So, but there are differences. So the trick has always been, how do we train T cells to recognize cancer? Well, it's been hard in the past. Limited success to immunotherapies, right? Until CRISPR. Now we've had remarkable success because we can be very precise about it. You take out a T cell from that patient's body, you know exactly what kind of cancer they have. You do the genetic test to say, okay, this is the little receptors on the cell because of what mutations are in the gene and so on. I'm gonna engineer this T cell to recognize that, and then I'm gonna grow a lot of them and inject that patient's own T cells into the body. I don't have a slide for it, but there was a remarkable case a few years ago in the UK. For the first time, gene editing has been used to completely cure cancer. It was a 1-year-old girl. She had very late stage leukemia. She was beyond all the meaningful blood marrow transplants. She already had few. They weren't working anymore. Her own immune system, in fact, was so low that she didn't even have enough T cells to try the approach I just described. So they had to do something even more radical. And they were allowed to do it because there's something called a compassionate exemption. So all these FDA rules and so on, they apply, but if you literally are in your deathbed, and this poor girl was 1 year old, they were giving her palliative care, which means preparing her for death. And they were allowed to try this one thing. So they synthesized T cells. They took other T cells that weren't her own. So they then had to use gene editing to tweak them so that they'd have the right little signals so the body wouldn't reject it. So that took gene editing. That had to change a bunch of genes. Then they made it have special little receptors to go after her leukemia cancer. Did a bunch of other modifications, injected her. She ended up cancer-free, and she's alive today. So I think this is where the promise is. And the first trials in the US are starting in Pennsylvania, very shortly, specifically using CRISPR. So this, to me, is where the promise is. Now let's talk about part of what we're here for, which is the risks and perils of technology. And I should have had one more fancy diagram, but I can just explain it to you all. You'll hear a lot about germline editing. That's the big debate, right? So let me explain that a little bit. It's fairly simple. If you did have some of your cells modified with CRISPR, and in your liver, for example, and maybe even we got to what's called stem cells in your liver. Those are the cells that create a bunch of other cells in your liver. Maybe we could have theoretically modified your whole liver, but when you have offspring, that didn't get passed on, okay? So we affected one patient. But what if at the embryo stage, an IVF clinic, Somebody has this $100 kit, this gene editing kit, and they make a change there. That's hereditable trait that they just modified, right? So if they get it right and they did something wonderful, this embryo was going to have a horrible genetic disease that was just cured, in fact, sort of taken out of the whole family tree. Perfect, wonderful. I don't think there'd be too much ethical argument about that. But what if you make a mistake that you didn't quite understand? Or what if it wasn't really a necessary procedure? What if a parent was paying you a lot to make sure your baby was intelligent, or had the right physical traits, or was the right color skin, or whatever else? That's where the ethical arguments really come in. So there's been tremendous amount of debate about what they call germline editing. In fact, Jennifer Doudna, the founder of this whole field, was herself tremendously frightened. She says in one of her books that shortly after unleashing this, she felt like Oppenheimer, when he discovered nuclear fission and then regretted that there ended up being nuclear bombs. She used to have nightmares, she said, that she was sitting across the table from Adolf Hitler, and he had his notebook out, and he was asking her, please describe this technology to me. So there's worries. She herself actually has come a long way, though, from a very frightened perspective to now sort of compartmentalizing it to think that there are very ethical applications for what they call germline editing, and some of these are being explored already. China, as you probably won't be surprised, is ahead of us and experimenting quite a bit, which leads to some of the other concerns later on the slide. But when it comes to some known horrible genetic condition, that's where the experiments seem okay. Health and safety, even some of those ecosystem comments I made, even the mosquito comment. That same study that's looking at eliminating diseases carried by mosquitoes by making them incapable of doing so, they've had open debates. Why don't we just eliminate mosquitoes? We could plug in a gene edit and plug in that little gene drive that makes them only produce males, for example. The entire species could get wiped out very, very quickly. If you all know math and know how that geometric progression works, that with a species that replicates so rapidly, that could happen very fast. And on the surface, that could seem wonderful, right? But then you start thinking about the unintended consequences, and humans have had plenty of examples of unintended consequences when we mess with something so dramatically, right? So bats, for example, all of a sudden, you probably made a lot of bats extinct, and then what happens when bats are extinct? Who knows what else they do, and so on. So the unintended consequences, even of some of these things that seem really noble, just have to be done very, very, very carefully. And finally, in 2016, there was a national security report in the United States that put gene editing as one of the top threats to national security, with the potential of creating weapons of mass destruction. And the concern particularly was it's cheap, it's powerful, and it's almost impossible to control. And it really is. If you have people all around the globe now proliferating this technology, tweaking things in bacteria and so on to see what happens, either accidental or intentional, there could be ramifications. So where there's great power comes great responsibility and great care. So one might think, hearing all this, all these wonderful things and then the scary things, where do you end up? On the balance, where should you end up when you have discussions with this, with your family, with your colleagues as it comes up? Again, it might not use the technical jargon, but I'm sure things related to this will come up. Well, here's where I come up. I think, first of all, I really think the promise far outweighs the peril. It really, really does. Plus, the genie's out of the bottle and we can't put it back, so there's that. It's here. So about all we can really do, I think, is just accelerate the good applications, be as wary as we can about the more frightening applications. There are, of course, global agreements that are happening about germline editing and so on, but the question of, which countries will really respect that and who's doing what in some secret lab. But the good applications are really astounding. Proof of that for me was in the time that I started making the slides. Just in less than the past 2 weeks, I just happened to glance at some of the latest and greatest news on CRISPR, and it's pretty phenomenal. It really is. So at UCSF, they successfully edited normal cells and turned them back into stem cells. We didn't get into that part of biology, but you probably all heard enough that when we start out, we're just one cell, right? The zygote has our DNA in it, that's it. And then you might scratch your head, how the heck does that turn into a muscle cell and a nerve cell and everything else? Well, you know what the answer is? now that you're all educated and bio-experts, it's actually those volume knobs. So at some point, the cells start getting the signals, and they say, let's turn on that gene, let's turn off that gene. And suddenly, that line of cells from then on becomes a muscle cell or becomes an eye cell. So the question, we've all known embryonic stem cells are incredibly powerful. You've heard for years their applications in medicine. They're like almost magical power. Here's the problem. We don't have any anymore if you're a grown-up, unless you saved them somewhere, right? And they're kind of rare in your body, even like a muscle stem cell. It's not quite the same as an embryonic stem cell. It's sort of a stem cell, but it's already kind of constrained. So getting embryonic stem cells is really tough. So just last week, a study comes out. Guess what? They use CRISPR-Cas to take a cell from any part of the body and turn it into the equivalent of your own embryonic stem cell. Here's what I found fascinating, and here's why I'm so excited about this subject. At the end of the paper says, we're not exactly what the killer app for this is going to be, but, pretty interesting. We know embryonic stem cells are valuable. Well, guess what? 2 weeks later, a paper from Stanford says, nothing to do with gene editing. They just said, hey, we were able to take embryonic stem cells, and we had a theory. Cancer cells, when they're really going crazy, look an awful lot like stem cells. In fact, one of the successful companies I was involved in was called StemCentrix and was operating just on that principle that the growth in tumors really come from the cells in the middle that are acting like crazy stem cells. They're doing they're spewing out all kinds of mutations, right? So these scientists said, gee, that looks kind of like an embryonic stem cell in terms of its outer signals. Yes, it's diseased and doing crazy things. I have a theory. I wonder if we could somehow train our own immune system with a healthy embryonic stem cell, if only we had such a thing, and inject it right near a tumor site and kind of see what happens. Well, the results were astounding. The results were your own immune system, unedited, unchanged, just doing its thing, was able to recognize that stem cell, train on it, and then suddenly all of a sudden see the cancer and go after the cancer cells. Well, now let's connect these two papers. I joked with my wife, Jamie, I wonder if these two people are talking yet. They should be. It's only a week apart. They will be, I'm sure, within the next few days, probably. But here you have one group saying, gee, with CRISPR-Cas, we can create an indefinite supply in any human of their own embryonic stem cells. Then you have another researcher saying, gee, if only we had embryonic stem cells from a patient, we just proved that we could train them to fight their own cancer. That's the kind of thing I think that's happening. That, to me, is one of the reasons why technology is worth pursuing, so exciting. Another study just came out past 2 weeks that proved autism, schizophrenia, bipolar disorder. Guess what? Not surprising, really. They're all diseases where they've proven it's abnormal gene expression, meaning the volume knobs are abnormally on in certain genes in the brain. They just kind of proved it. They identify which knobs. Guess what? Now we have the ability to potentially tune those knobs. The CRISPR, some slightly wackier but very interesting applications, again, just in the past 10 days. CRISPR was used to create a molecular DVR in cells' own DNA. So what that means is, remember how long I told you DNA was, right? And there's a whole bunch of it that we're not sure what it does. Think it has to do with the volume knobs, probably other important things, but there's some of it that clearly seems pretty useless, just kind of repeats and doesn't do anything. What scientists have been doing is having CRISPR-Cas in the gene. It creates molecules, they tune it to create molecules that float around the cell and act as little chemical sensors. If they see a certain protein that's a sign that a reaction starting that looks cancerish, for example, they record that right back in the gene and right back in the DNA. They squirt it right in, edit it, put it right in. Maybe they attach a atom that's a phosphorus protein that glows when the scientist puts something on it. Next thing you know, we're looking at lab cultures and we're having certain cells glow. Oh, that one's starting to be cancer. That one's starting to be cancer. Crazy applications. And then finally, Jennifer Doudna herself, just last week, published a paper. What she did is she took this incredible fidelity that the little CRISPR machine has for detecting and identifying unique pieces of DNA and said, hey, what about viral tests? We have some that use a different technology, kind of expensive, kind of slow, not very high fidelity. So you can tell, certain kind of viruses are there, but some viruses have a gazillion subtypes. Some are really dangerous, some aren't. And we don't have that fidelity yet. Now we do. She used CRISPR-Cas to create probably the world's most accurate, cheapest, fastest, and highest fidelity viral test just last week. So to me, I think the genie's out of the bottle. We can never put it back. If you really extrapolate, you can let your mind wander. Just about every sci-fi movie you ever saw is probably going to be possible in any of these things for good or bad. But honestly, if human beings are going to live longer, which we undoubtedly are, if we're going to put more stress on our resources, if food supply, planet, and so on, we're clearly heading for a train wreck if we then don't have the ability to try to mitigate or help some of those other things that we affect. I've always been a believer, think even, use a Montana example, if you're a hunter, game management, right? I've always been of a theory, either you leave the whole system alone, let it reach its equilibrium, or once you start managing anything, one species, you better be managing most of the rest in that food chain too, or else you're going to have chaos. Well, that's kind of where we are as a human race right now. In fact, in dealing with life itself on our planet, as we can now clearly manage ourselves a little better, we can start to manage other things. It's, in a way, potentially a good thing that we have the ability to tune other things that start to get out of whack, mitigate our negative impact, and so on the climate and so on. But that's where we are, and thank you very much.
If you won't mind to keep your questions brief, because we have about 300 people in the room and we want to get a chance to get a lot of questions around.
Thank you very much. This is fascinating. I'd like to bring it home a little bit. We have, of course, a sizable medical community in the Flathead, and it's a sizable part of our economy. Two-sided question. One, how will this affect the day-to-day practice of medicine here in our community? And then the other side of the question is, how might our medical community here contribute to this science?
I think the answer to the first question in the nearest term will most likely be some of the discovery innovations, testing innovations. So one of the arguments made about the real power of CRISPR is not only does it allow us to alter these incredibly intricate, critical little machines of life, code of life, but also to understand it. So that little molecular DVR, by the way, the killer app for that molecular DVR, I gave the cancer example, but one of the ones they're most excited about is, what's one of the areas we're most still in the dark about in biology? It's our brain and neurology, and really how do thoughts happen, how does our consciousness happen, and so on. They're thinking about using that technology to embed it into neurons, so they're actually recording events that they're experiencing, and then we can kind of play it back and try to figure out what's happening. So I think the advancement of the science and understanding will have sort of this fairly rapid, continuous, indirect impact on practices and so on. I think you'll have a direct impact in terms of the availability of some of these things like the testing and so on, that will just make things more accurate, quicker, cheaper. Again, we shouldn't underestimate the fact this stuff is so inexpensive kind of matters, right? What's it take for mass adoption is something to be super simple and cheap, and that's one of the elements of this technology, which makes it both powerful and frightening. Second, you said in the Valley, what would be the longer term effect, or how could we? Well, we could start starting a lot more neat little technology companies or biotech companies. I'm already involved with a couple of my best founders that are sitting right over here involved in a genomics company that's helping actually speed the process of discovery. I didn't get into it here because it was kind of a little off tangent, but to me, the urgency of understanding the rest of that DNA is kind of as high as it's ever been, because now we have people tinkering with it. So it'd be kind of nice to really understand it as fast, for the world to really understand it before the tinkering gets out of hand, or we know what to tinker with. So the problem there is the cost of genetic sequencing. It's kind of an interesting little side story. The Human Genome Project, if anyone's ever heard of that, that's what started a lot of this. It was over 10 years and about $3.5 billion to sequence, get all those letters of one human genome. Now it takes minutes to hours and around $100, right? So the amount of data has gone through the roof. We're sequencing everything, right? The problem is that you get into a pretty complicated computer science topic if you have terabytes and terabytes of data and you're looking for a needle in a haystack, and oh, by the way, you're a PhD in biology and you don't really want to understand computer science and the 65 different programs you have to knit together to get your result, and oh, by the way, the paper you're trying to replicate weren't really clear on which programs they use, so you're getting different results. I mean, that's a problem. What it means is we really only have this elite part of the ecosystem, the very best universities, the very best well-funded bio companies with the $150 billion market caps. We have those guys those folks making progress. But what about all the other human brains that could be working on it? So that's what this company XD Bio is up to right here in the Valley, is helping democratize all that. That's why I love those guys. They're doing such a good job of making it so that, right down to the freshman bio student, you could start doing experiments with sequencing data and understanding. So, I mean, my long-winded answer is I think a lot more technology companies playing in the space and then having synergies with the health organizations.
Hello, thank you for being here. This is amazing information. I've just came from a blockchain convention, and I've been opening up the idea and the concept of that, everything that you're speaking about, and that we are actually the blockchain, and that our DNA holds so much technical data that we're essentially, all the different energy and sounds and lights and everything can turn off different codings inside of our DNA. And if that's the case, how long is it to where we basically become the technology and we're the machines?
Last week. So there was another paper I didn't mention because there were so many to mention, right? But really amazing, kind of somewhat crazy things are happening. There was another paper, and I believe this was East Coast, might have been MIT. So much like this gentleman was just talking about, they looked at DNA and they said, you can encode a heck of a lot of information in DNA. It's much better than my little, most expensive little, however many gigabyte little SD card or whatever. So they did an experiment. They literally encoded a digital movie. I forget which movie it was. I think it was a horse galloping. They then translated it, made up their own translation of what the ones and zeros should translate into, A, T, C, and G. Then they used CRISPR in a bacteria to put it in that bacteria's DNA. Then they let the bacteria replicate millions and millions of times. Then they picked out another random bacteria and were able to extract and replay the movie. So it was the ultimate, distributed storage application. There's, who knows what it's good for, but yes, is the answer.
Hi there. Is there any potential that somebody could use CRISPR to create a CRISPR-resistant cell?
I believe so. I think just about anything is possible. Yeah, and by the way, let me tell you that the pace of progress is so fast. There are certainly bumps. So one of the bumps was, it's an irony to this too, is that despite the precision of the CRISPR little targeting mechanism when it slices up DNA, remember those prior technologies had a couple of % success rate and they did collateral damage. So we really had nothing, and this is oftentimes, if they do the experiment right, potentially way up in the high 90s as to that precision. But occasionally, they've discovered it did make some errors. It would find if there was a code that almost exactly matched the target code, maybe it was off by one letter in a lot of letters. There were times when CRISPR, the little CRISPR, at least the early versions of the mechanism, would say, it looks close enough, and cut it. So hence, worries about danger use in humans. That's actually why, and here's the irony, that's actually why, ironically enough, these germline experiments, when you're taking like an embryo, while they have more ethical considerations and more potential dangers, they're actually easier. So the outside the body applications are easier and are kind of progressing faster just because those very reasons of when you go inside the body, you darn sure better be right. Now, by the way, there's a bunch of government money that just was allocated to fund the advancement of CRISPR, and they're tweaking the enzyme, the little scissors, they're tweaking a bunch of things, and sure enough, it's getting more and more and more and more accurate. So it will come, but there definitely have been some setbacks.
We have a very young person.
I'm in 5th grade. Oh, yes. If I input a different gene, could I change something like my eye color?
Theoretically, I guess, certainly if we did it when you were one little tiny cell, we could do that. I don't know enough about how fixed it gets once you're running around young lady in terms of when the change would take place. But yeah, there definitely is a gene that determines eye color. It's just a question of when does it kind of get cemented in place. Anything else?
I can just ask you without a microphone. Is there any agenda to do a backup and restore in case you really screw up?
You know, gosh, I figured I had so many pictures of my slides, it wasn't worth it, but I have a great picture. I actually bought it from Shutterstock, so all these pictures, I didn't want to worry about the little copyright thing. But I had one actually for that topic. So after that report came out from, I think, the National Security Agency in 2016, I believe that was the agency that listed the top 10 dangers to the United States and listed gene editing, CRISPR-Cas as one of them. Shortly thereafter, there ended up being a program funded by the US government, and there's a paper with a neat diagram that's all about ensuring that we build in reversal mechanisms. So, if you're going to be playing around with the germline and you happen to screw up, or a scientist somewhere happen to screw up, let's make sure we develop the chaser technology, that can kind of chase the edit down and try to reverse it. So there's a lot of thought being put into that.
Thanks so much for the excellent presentation. What conflicts do you see in this level of science with religion?
Well, it runs the gamut, because remember, there's folks who have religious beliefs that it should all be up to God, and you shouldn't go to a doctor. It's God will determine your fate, and therefore, when you're sick, it'll happen or it won't, right? So you have that end of the spectrum, and then you have, of course, the incredibly non-religious, 100% science. And then you have all the in-betweeners, which probably most people in the world fall somewhere in between that spectrum. I think the ethical considerations start to become entwined with the religious considerations when we really start talking about these, what they call designer babies, that we should be able to just design these, frankly, sort of superficial, trivial traits just because we want to, and so on. I haven't heard as much religious argument from folks when we're talking about some, horrific inherited genetic mutation that's going to cause the baby to have some horrible condition, and can we fix it? At least I haven't heard the cogent argument on that side linking to religion, but yeah, it's an element in the debate, that's for sure. I mean, step back for a second. We now have the potential, human race has the potential, after life's been on the planet for 3.5 billion years, now in the last couple of years, we've gained the potential to completely change the evolution of every species on the planet if we wanted to, right? That's the debate. Yes? Sorry, yeah. You need a mic?
What are the implications for our healthcare system, say, 10 years out, 20 years out?
Oh, you picked a nice long target, because I thought you were going to ask again in the next year or something. But I think it's profound. I think it's really profound. I think, as I was describing, and as you can do your own research on, you'd be really hard-pressed to find a dramatic illness or a life-ending illness or life-changing illness that doesn't come down to, at its core, something going on with the DNA or gene expression, as they say, the volume knobs. And now we have a tool to manipulate that. So I think you're going to see profound progress in areas that just have been stuck for so long. I remember even cancer, and this is what frustrated me when I started getting into the bowels of cancer research with some of the brightest folks in the Bay Area in California, where a lot of the great biotech companies come, is I was just astounded by the almost medieval nature of the care we had. And there's no criticism to anybody. I'm just saying, when you really think about it logically, here's this mechanism that's causing the tiny little molecular machinery in a cell to go a little crazy and to spew out these little cells. And since we had no way to get at that, we're just trying to think about these outside things that maybe would help. Well, maybe if we give this dose of massive chemicals that basically kill every cell in your body, because the cancer cells are growing faster, maybe it'll hurt them more than the cells that are growing a little slower, but still killing every cell in your body, for example. I mean, to me, that reminded me of bloodletting, in the old days, or when you had a migraine in the 1700s and they drill a hole in your head to relieve the pressure. You know, I think we'll look back at it like that. And again, it's not criticism of where we are today. We've had tools that we had, but now that we have these more precise tools, I think you'll see some real innovations. The immunotherapy is really exciting to me. Using your own immune system and just powering it up. Go ahead.
Do you have an application for CRISPR with prions?
Creons, you say?
Prions.
I know a lot about a lot of things.
It's like mad cow disease.
Oh, yeah, yes, absolutely.
Prions.
Right. Yes, I mean, I bet, I haven't done the Google, but do a Google CRISPR prions, and I bet you'll find a hit. I'm serious. I bet you will. Because, yes.
Oh, we have a microphone here.
What do you think is going to happen if all the diseases start getting wiped out? Is the pharmaceutical companies going to go away quietly?
You just said what are the pharmaceutical companies going to do when diseases start to go away? You know, I think despite all this wonderful technology, I think we have a path to get there someday, but you know, it's probably still going to be quite a while. I don't know, maybe pain relievers for when we break our leg skiing, or no, I'm kidding. I think you know, a heck of a long time before we could have wipe out all human disease, but you know, it's a statement that's probably truthful to say we can now credibly see a time when that could happen someday. You know, I think we've gotten to the. Hopefully, if you've gotten anything out of my talk, you've gotten the point. We've gotten to the source of everything. We've gotten to the ultimate source code of life. That's kind of that dictates everything. Remember, you know, that little strand with its couple of chemicals that come along in the egg and the zygote unfold to become you. You know, it's a pretty sophisticated piece of machinery, right? And now we're down there tweaking with it. So,
this is kind of an ethical question. You mentioned some ethical dilemmas in altering someone's appearance, whether it's skin color or speed or intelligence. From your point of view, what would be unethical about that when there's parents that can choose to, who might have a inherent disease and they're not prohibited from reproducing? Wouldn't this be a very similar aspect in that?
You posed a couple of things. I'll tell you the morally clear examples, at least all I've heard, are our cases like, you know, there are parents when they get that genetic test and the doctors say, I'm sorry, when you have a child, you know, you have this recessive gene, you have this recessive gene for this bad thing, and that child's going to have it, kind of, no doubt. And they're told either have a child with this horrible affliction or don't have a child. For those parents, we now have a solution, and I think that one's one of the more morally clear examples. But you know, when I've used this reference to the genie being out of the bottle, we can talk about all these moral, what do you call it? You know, speed bumps that try to slow this down. But I think we're all pretty intelligent. We all know and can guess 10, 20, 25 years from now what this is all going to lead to. I think it'll lead to a very different world. I don't know why or how we could restrain, for example, once intelligence is fully understood. And by the way, intelligence is one of the more complicated genetic-related expressions. There isn't just one smart gene. You know, there's, as with a lot of things, it's a combination. It's this gene and this gene and this gene being overexpressed this way and this way. So it's a formula, but it's still all genetic. When that gets a little more understood, I almost don't see how, from a self-evolutionary point of view, human beings with knowing how to tweak that wouldn't someday start tweaking that. And then it gets a little interesting, right? Because then we get more intelligent, and then we get more intelligent about the use of what we can do. That's where you get into sci-fi. You know, for any of you computer geeks, there's Kurzweil had this projection. If you're a computer person, you'll understand this. Most of you probably won't. But ever since computers were invented, there's something called Moore's Law, which meant that the speed doubled every year and a half, I think. And so there's this crazy exponential curve as computing power got more and more and more powerful. And there was this sci-fi kind of philosopher, Kurzweil, who projected that when and if we ever reach the point that the computing power is more powerful than the combined intelligence of all the human beings on the planet, then like all bets were off. You know, very crazy things happen. I kind of feel like we're there now in in evolution and biology and our ability now to do what we can do. I think you could easily see a genomic singularity point, as Kurzweil called it, where, wow, again, kind of along the lines I talked about, what if we make ourselves down the road, 100 years from now, make ourselves more intelligent, then we understand more about how to make ourselves more intelligent, and then we make ourselves more intelligent, and who the heck knows? There are some Elon Musk-ish kind of practical examples here. If he wants to colonize Mars, again, ethical considerations aside, I'm talking maybe long past all our lifetimes, but who's to say we won't be tweaking the genetics of people who are going to spend their lives on Mars or another planet so they can adapt a little better, right? Can handle less oxygen, higher heat, whatever. I mean, all bets are off. There are bacteria and living creatures with DNA that live inside volcanoes, right? So that says it's possible. Remember, we all have the same code. Theoretically, we could turn one of your cells into one of those if we tweaked all the right knobs, right? So. There are no stupid questions. Didn't you ever hear that?
But given, just by thinking about this, given the ramifications that could happen, why was it given to the public?
You mean like the little do-it-yourself kits?
Yeah, why?
I don't know. I was a little surprised by that. I think, frankly, just like in the cartoon, the genie getting out of the bottle and the vapor dissipates so quickly, I think it all happened so fast. I think if you listen to Jennifer Doudna and some of her speeches, who again, she was one of the ones who was so thrilled and so determined with the moral righteousness of what health problems she was trying to solve, yet she's also one of the ones most frightened when the genie got out of the bottle and proliferated so fast. I think it just happened so fast. And, I remember reading something about how instructions for germline editing, we're about to go out, and a whole bunch of other scientists got together, and we're going to put them out on the internet, got together and talked that publication into not publishing the paper. Just kind of like the recipe for a nuclear bomb. I'm sure there was pressure to not get it out there, but I bet in some weird corner of the internet, there's probably the recipe, right? So I think it just happened so fast. Ben.
So is CRISPR, is it hardware based or is it a process, and is it owned? Is there
well,
intellectual
funny you should
property
ask,
there?
because if any of you have dug this deep into it, I highlighted Dr. Jennifer Doudna, and she is kind of most recognized as sort of the inventor of it all. Not everyone would agree with that, including one of her former PhD students. There's been a wicked patent war about who really published their paper first on CRISPR, and it has to do with, and they each have companies, so she's associated with a few companies. They're all clustered around MIT, like really right down the street from each other, but they're battling. And I don't know, and I haven't followed it, but yeah, there's some debate as to who kind of owns what. But so far, in terms of the applications, there's been free to use, free to experiment. I mean, given to the scientific community, which kind of means everybody, especially with these do-it-yourself kits. Literally, in Googling some of the papers that I wanted to look up, in terms of progress on diseases, the ads pop up at the top, the sponsored links. Some of the sponsored links will get your own CRISPR kit, Yes.
I have type 1 diabetes, and I was just wondering if gene editing could possibly provide a cure anytime in the near future?
Yes. Yes. So she asked about type 1 diabetes. So again, there were too many recent breakthroughs or advances using CRISPR to list. I made one of my slides too busy already, just listing half a dozen, but it could go on for a number of pages. And I was specifically reading one about, I believe it was in a mice model, they call it, right, where they model human pancreas, and they tweaked the gene in a diabetes patient, and they were able to restart the correct, they were able to completely reverse diabetes and restart the correct insulin production.
Given that we can synthesize different things, it would seem that we're running into a sort of human-esque, like Archimedes ship kind of situation. So, like, where does it get to the point where humans aren't human anymore, but simply just people? And also, sorry, kind of two-part question, is there any sort of UN-based rules and regulations around CRISPR in order to stop countries, not countries like America, but other countries that are less governmental based, to stop them from doing things like eugenics-based genocide?
So, Dr. Doudna herself, as I said, I mean, as you can tell, tremendous respect for this woman. She not only was the inventor, she was the first to be frightened by her own invention. So she led the creation of the first international set of meetings, which have now continued, and I think they're huge now. But the first one was shortly after the original invention with 20 key scientists. And sure enough, international regulatory bodies are passing rules. But as you rightly pointed out, this isn't building a nuclear bomb, which required however many billions of dollars in crazy labs and so on. This requires a couple $100 kit to start playing anyway. So, and literally a fairly sophisticated lab for dollars that would probably be in the thousands, not in the millions. So the question of, can we, what can we enforce, and what can we, is a real one.
How do you think CRISPR technology will affect the community of genetic counselors or people who look at people's genes and see how predisposed they are to previous conditions? I
mean, I actually think that'll have a very positive effect. It'll be more in the understanding. I mean, I really wanted to make sure you folks understand, as mind-blowing as all this is, I really hope you end up, and this is fairly critical, I hope you end up with your meter sort of tilted to the right side of it, which is, I really honestly believe, the promise really has to get weighed against the peril, but there's no putting the genie back in the bottle. We have this now technology, so the question is it morally right to use it, or is it morally right, which is the greater moral sin, to use it with some of the risks that it has, or to know we could use it and to not use it in a case where there's some severe human suffering that we know we could do something about. So anyway, I mean, yeah. Go ahead. Did I forget, did I hit on the main question there?
I was kind of thinking more job outcomes, overall positive growth, or
Oh,
declining
I know.
growth.
You were talking about genetic counseling. Yeah, I'm sorry. I get up to that critical tangent because I just want to make sure we talk about such scary stuff. I don't want people walking out of the room and next thing you hear, whenever you hear anything about this technology, it's a no, vote no, ban, suppress, whatever, I'm going to go hide in my closet. No, I think the most sort of subtle but dramatic impact that we'll start to see, and we've already seen it, frankly, is on the information side. Because as I said, just think about it really simply. I can't even imagine how we were discovering some of the things we were discovering about genetics before this. Because, I mean, if you want to know what a gene does, but you had no ability to turn it off or on to see, it was pretty tough. So credit to the scientists that were making the progress they were. But now we can do things a little more precisely and logically and faster. You can, in a very rapid time, fairly cheaply, if you're in a good science lab, you can take a plant you're looking at or whatever and start looking at all the genes. What does this one do? What does this one do? And so on. So even in the genetic counseling, I think we'll get past any residual, junk science that may have been around for a while and get to real answers.
Hey, Michael, excellent presentation. I have a question. Should I cancel my cremation plans in favor of being frozen?
It's really funny you said that. That's a serious question because I've always had my father, who we love very dearly, passed away recently here in Whitefish, and he did what I had always dreamed about. We cremated, ashes out in the forest, sprinkled around, and all that good stuff. And, you're part of the forest forever and everything else. And then I read one of these papers that said, DNA can last for a really long time as long as you're not cremated. And I went, oops.
So I have two questions. My first question is, what is the largest barrier that you see for the advancement of this? And I'm not just talking about I'm kind of talking about like the everyday person, because as you've mentioned, we all have these ideas that potentially it could head in really bad directions. So education is important. And what is being done for some of the pushing or the proper education of society? And then my other question is, do you know what institutions in Montana specifically might be focusing and helping spur growth in this area?
Well, in the understanding, I mean, maybe we're starting here, right? Up in Montana, I agree completely that it's critical. Hopefully you've all agreed after listening that, gosh, you really better understand this stuff, because, if you didn't and you just heard some scary one-liner about it, oh my God, there's those scientists, tweaking with things and wrecking life and organic veggies and all this stuff, right? You'd think bad things and you'd vote against it and so on, and look at the potential good, that you could be preventing. So I think education is key. I'd love to see a lot more happening, around Montana, infused into some of the curricula and so on. It's all pretty new. That was a real data point I gave you when I said, I was surprised myself when I literally, from the time I started thinking about putting these slides together till today, I Googled the latest news, and a lot of that incredible stuff was literally in the last 10 days. So the pace is really incredible, like nothing I've ever seen in science.
So with all this technology, do we get to the point ever where our personal choices don't matter, where we can just live a reckless lifestyle because we have this ability to turn off or turn on genes, that it doesn't matter what choices we make?
That one might be a ways down the road, but I guess theoretically possible. I will mention one thing that I've gotten most really excited about as the pieces start putting together, and it affects us a lot here at home, Flathead Valley. Which I started thinking as I was getting into all this. Gee, I wonder. It would seem to me that addiction, drug addiction, alcohol addiction, would have to be related to this, right? I wonder if anybody's looking at it. And I was actually somewhat surprised. There's research that, guess what, indicates from about 10 years ago, way before all this stuff, way before we understood a fraction of what we understood about gene signaling and everything, that said, gee, addiction, when we looked at the neurons in addicted people, they seem to have some unusual patterns in gene expression. I mean, why wouldn't somebody be looking at that now? Because what better solution, let's say there was, just hypothesize for a second, because I think it's going to happen. I'd love to help it make it happen. What if there ended up being a safe pharmaceutical that did pretty much exactly that? You take an addicted person, and we all know about addiction and how incredibly hard it is to get that monkey off their back, and it isn't just deciding to do it and so on. It's chemical. It's bound to their molecules, and that's really what I think it is. And I think any scientist who digs into it would say, well, of course, now that totally makes sense. Is what happens is these poisons you've taken from the outside have affected the signaling of your genes, and they happen to be the sticky signals, right? The ones that kind of really stick on there in the wrong places, and they just are really hard to get off. So what if there was a pharmaceutical that reset your knobs back to where they're supposed to be? Now, sure, you probably didn't prevent you from getting addicted again, but if you had the chemical part, the molecular part of the addiction gone. So I think that's fascinating. It's super worthwhile.
Just a question about, it seems like because what you're describing is so discrete and there are, known reactions, and given the scale at which this happens with molecules and at that scale, that there'd be organic computing possibilities. I mean, if we had these reactions, if they happened at a fast enough rate, you could control things to literally, make a Turing-complete organic computer out of these type of reactions. Is anyone ever looking at that at this point?
I'm not sure. I did have a bit of this guy obviously knows technology, so delve into geekdom for a minute, because I was a geek for so many years. I did have a sort of an analogy that struck me, that, if anyone knows anything about sort of the history of computing, it did start with the ones and zeros, right? Sort of electrical signal, hey, I can make it turn into 1 over here if I apply a charge and 0 if I don't. Cool. Then somebody put together those things in a little pattern, said, gee, now I can put together what's called a gate. If I put a 1 in here and a 1 on here, a 1 pops out. I'm going to call that an AND gate, right? Because they both have to be true. I'm going to do an OR gate. I flip them around a little bit, if one or the other is true. With these little building blocks, we have the iPhone, you know, eventually, right? They're all stitched together, then I can do math, then I can do it, so on. I had the same thought with CRISPR and the progress so far and where you could go. What if you could actually start to construct incredibly complicated if-then-else statements with CRISPR? And I think you can. I actually think some people have already beat me to it. There's something that I'm reading about, sounds like they're doing similar, meaning it isn't just go in there and look for a match and do an edit. Why can't you have conditionals? What if only if this is a match and this is a match, but not this is a match and that one's mutated, then change this other gene way over here? I think the way that these little molecular packages can be, you know, suitcased up and packaged and only unwrapped if this condition is true, I think we're getting there. So that leads a little bit more to very complicated. I actually think there's going to be a molecular programming language. Because, by the way, that iPhone picture I showed you, for anyone who's not technical, that's not how people program. I was a little misusing the term when I said source code. That's actually called machine code. And source code is something that sort of looks like English, you know, has lots of weird symbols and stuff, but at least it's words, right? And right now with genomics, you know, we're dealing with T's and A's and G's and so on. So I bet there could be lots of advances in fairly sophisticated ways to describe what you want to do. Some of it's already happening, but I bet it could go further.
Hi, I'm a psychology and social work major, and so a lot of times we talk about how anxiety and depression are like both biological and psychological and involved with the emotions in the brain. So I was curious on the last question about addiction and stuff, like how much do you think that we just don't know about the brain and those kind of things, like our emotions and how we respond to problems in our lives are something in our genes, or that it's something more that, you know, can't be scientifically, you know, fixed, that it's just a part of us and humanity?
I think it's a great question. And the answer is, I didn't mention it, the same study that saw the abnormal gene expression for schizophrenia, autism, and bipolar also noticed some abnormalities with patients that were severely depressed in gene expression. So this is the way I think about these incredible little machines that govern all life, right? Just that little protein machine and on and off switches, that's all, right? And somehow we become us. I would think about it this way. There's a lot of those on-off switches that are happening all the time, right? And they're supposed to, fairly frequent on-off switches. Hey, I'm in a bad mood. Hey, I'm in a great mood, and so on, right? Where does that come from? What does that mean? It's probably because little genes were signaled a little bit differently, causes a little bit different reaction with the neighboring neuron, right? Based on a signal you sent out from the DNA. That's where all the signals are coming from, right? You're spewing out molecules all the time. But then there's other cases where it gets abnormally stuck, you know, or a different gene gets turned on where the switch is a sticky switch, right? So I think it's a combination. I think there's a lot that happens every single second that we're breathing. You know, remember 10 trillion cells, so there's a lot of stuff going on, right, in each of those cells. But then there are cases we've found that the persistent conditions that affect human health are related to little malfunctions, right? Anyway, thank you so much. Really appreciate it.
Thank you.